Good morning, accelerated biology students. Mr. Adair here. We're going to start week 3, and we're still learning about cell division. Hopefully, everyone had a great holiday break. So, I'd like to go through a couple things for week 3, and for now, you can just follow along. All of this information, as you can see here, is in our Schoology folder. The first thing you need to do is go back from last week and think about the 4 reasons why cells divide and write those in the discussion post to earn points for the day. And then the second thing you need to do is open up the cell cycles PDF, which that is what I'm going to go through right now. And this will be information you will need for a project we'll be doing this week. So 1 second. I'm just going to leave it like that for now so I can still be on here. Okay. And I'm going to go through this a little bit quickly because you also have this information, but we're learning about the cell cycle, that's cell division, which is really more like multiplication. When cells divide uncontrollably, we call that cancer. We've learned about that. But today we're going to get into the actual, basically the stages of how this works and talk a little bit about chromosomes. So after today, you'll understand the main stages of the cell cycle. We talked a little bit about this last week, and this is the diagram going through the cell cycle. This is going to be an important piece of information for your project. It goes through interphase, which is G1, S, and G2, and essentially that's when the DNA replicates itself. And then we have mitosis, which consists of prophase, metaphase, anaphase, and telophase. One way you can remember that is the acronym PMAT, P-M-A-T. Then the last part is cytokinesis, where the cell actually divides. So the first part, G1S and G2, is going to be considered interphase. And the cell grows, replicates the organelles, makes a copy of itself, kind of grows and gets ready for mitosis and then division. Now, this will be kind of the first introduction to chromosomes. We've talked about them a little bit, but during this interphase is where the chromosomes will duplicate or double. And for your project, you will be following the path of 4 chromosomes. But I want you to know, in reality, in a human, we have 46 chromosomes in our body cells. So that means every one of your cells, your skin cell, muscle cell, nerve cell, inside the nucleus, you have 46 chromosomes. We'll learn later this year where you got those chromosomes, but real quick, you got those chromosomes from your parents, 23 from your dad and 23 from your mother. So you have 46 chromosomes, but for the sake of our project, when we talk about it, you'll track 4 chromosomes only. All right. A quick review, what is a chromosome? A chromosome is DNA all wound together in combination with proteins. So what chromosomes really are DNA, and DNA is the code that makes your proteins, which makes you. All right. When a chromosome doubles itself, getting ready for cell division, it makes a copy of itself, and it kind of looks like an X, but it's really like one chromosome attached to another, and they're an identical copy. And we call those sister chromatids, which you have one chromosome here attached to another chromosome, and that'll make more sense when we go on and watch how the chromosomes need to divide. Okay, so mitosis is after the beginning interphase, and this is where the nucleus totally divides apart. It happens in every single one of your cells except your sperm or egg cells, and it creates genetically identical daughter cells. So if a skin cell of mine is dividing right now, it's going to create an identical copy of that skin cell. So overview, here are the main 4 parts, like I said earlier, of mitosis: prophase, metaphase, anaphase, and telophase. And in a normal human cell, we'd have 46 chromosomes. But in this example, we're going to track 4 chromosomes. And it's kind of hard to see this picture a little bit, But in this original parent cell, we have 4 chromosomes, 2 red and 2 blue. Then if you look in prophase, you can see kind of like an X. That represents the chromosomes making a copy of themselves. Then metaphase, they line up along the middle. Anaphase, the chromosomes start to pull apart. And then the last phase, telophase, right before cytokinesis, we have 2 new cells forming, each with 4 chromosomes. And if you remember, the original cell had 4, that's because we create an identical copy. Let's look a little bit closer into these phases, and this is what you're going to use for your actual project. Interphase, G1S and G2, the cell copies its DNA and it's getting ready to divide, okay? Prophase, the chromatin condenses into chromosomes and the nuclear membrane starts to break down and these spindle fibers form and they're going to start to go to opposite ends of the cell because the chromosomes will get pulled. You can use this information when I tell you about your project in a minute. Metaphase, I always remember this because the metaphase plate, it's like the middle of it, it's like the equator. The chromosomes line up in the middle and they're getting ready to get pulled apart. Now, we have 4 chromosomes in this picture with their sister chromatid attached, which is an identical chromosome. So we have 4 then with their attached sister chromatid. All right, anaphase. Anaphase, the sister chromatids start to pull apart, okay? And then telophase, the new cells are beginning to form. The nuclear envelope of the new cells are beginning to form. And as you can see, we have 4 chromosomes, 1, 2, 3, 4 in each of the new cells. And then cytokinesis, the final division, the cell completely divides apart. And in this case, we have 4 chromosomes. In a real human cell, you'd have 46 in each, okay? And there's some animations you guys can go do that track through this that can help you with your project. Okay, now going back to our folder, that's where you can find the PDF. So now your job is we haven't done this yet, I actually want you to physically draw out these steps and make a mini poster, okay? I made a very clear rubric for what needs to be on your poster. And this poster can just be a piece of printing paper or even like if you have a piece of notebook paper, you can do that. And what the first thing you're going to do with your paper is you're going to make 6 squares because if you think about it, we're going to do interphase in 1 square, prophase in 1 square, metaphase in 1 square, anaphase, telophase, and then cytokinesis the last. The best way to make 6 squares is to take your paper, I'll try to make this as easy as possible, fold it into thirds, and then fold it again, so we have thirds, and then you're going to fold it down the middle, okay? Which I already did this, but if you can see, I have 6 squares. If you want to draw out your squares because you don't like folding, you can get a ruler or just use your best judgment. And I'll just do this quick. 6 squares there. All right. And then, like I said, each square needs to be labeled. So let's see if this works in the camera. We have interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So you need to use your notes and the videos and draw out what is happening in each of these phases. And if you look at the rubric, you'll see, make sure you put your name and title and date and hour on your poster, fold it into 6 segments. Those are what needs to be in each segment. Then for the main points, or part of the main points, it's easy to get the first points by just doing the basics. Start your original cell with 4 chromosomes, label how the chromosomes go in each of the phases, and describe what is happening. And you can copy down what I did in my slides or watch the videos. And then to get the full amount of points, if you can make it look really nice by drawing it and coloring it. Now, if you don't have any colors at home whatsoever, why don't you let me know and we can figure something out for that. So technically this poster assignment is due on Saturday. That would be Saturday coming up here. I can't remember the date on that right now, but I'm giving you Monday and Tuesday this week to finish it. So maybe try to get that done and then take a picture of your poster with your iPad and then submit it to the assignment. And so that's what you need to be doing today. And I will talk about it in class. Period 1 will meet Monday, tomorrow. I'm doing this video on Sunday. And period 4 will meet on Tuesday. But that's the basics for now. And have a great start to the week as we learn about how cells divide and actually get into the physiology of how that works. All right, I'll talk to you soon.